So operon, what is operon? What you can see here?
There should be one promoter.
One promoter and many
Structural
genes. When one gene is controlled by, when many genes are controlled by one promoter, it is called as operon. Now here we have to study what is lac operon. What is lac standing for here? Lac, lactose. Okay, lac stand for lactose. Now these are the different parts of the operon, lac operon gene. So whenever if question asks, start your answer by drawing this diagram. P for ZY. P for promoter, I for inhibitor it is. Okay, and that is the regulatory gene. Okay, this is the regulatory gene. Okay, this will regulate working of these genes. Okay, this will regulate, this will inhibit these genes, or this will activate these genes. So we have one promoter, one regulatory gene. Then we have promoter, operator, Z, Y, A gene. So every gene, for every gene there is a promoter. Here we have regulatory gene, one promoter. Here we have three genes. For these three genes we have one promoter and one operator is there. Now lac operon consists of a regulatory gene or an inhibitor called I. Okay. It codes for a repressor protein. So this one, this I will give one protein. That protein will go and bind on this operator and will make this inactive. This regulatory gene, which is inhibitor gene, produces one protein. That protein, when it is produced, will go and bind on the operator. It goes and bind on the operator and it inactivates working of these three genes. Then we have three structural genes, Z, Y, A, and one operator region. We have already seen the function of these three genes, Z, Y, A. Okay. So role of the structural genes is the Z gene codes for an enzyme called beta galactosidase, which will break lactose into glucose and galactose. This beta galactosidase, it breaks the lactose to galactose and glucose. Actually, glucose is the source of energy, but in absence of glucose, if you give lactose, this gene will get activated and it will break the lactose to galactose and glucose. Second, Y gene codes for permease. It causes the permeability of the cell to lactose. Okay, means this Y gene will code for protein which will make the cell membrane soft so that it easily allows lactose to enter the cell. And last gene is A gene which codes for trans-acetylase. Okay, not much is given about this. This is just transfer of acetyl group, trans-acetylase. You all read at least act as a trans-acetylase. Then next is what we have is the working, how the regulation of gene expression takes place in lac operon, the working of the lac operon. Now, in the absence of inducer, the inducer here is lactose. Only when lactose is there, then this entire lac operon will work. If there is no lactose, there is no need of this gene to work. Only if there is lactose, this lac operon will work. If no lactose, no need of doing this gene, no wasting energy in doing this work of making this beta galactosidase. What is the function of beta galactosidase? To break lactose. If no lactose, no need of any beta galactosidase. So in absence of lactose, that is inducer is lactose here. In absence of lactose, what happens? This regulatory gene, no, it will transcribe a mRNA. It will undergo transcription to produce mRNA and it produces a repressor protein. This protein will go and bind on this operator and there will be no structural gene working. This much only. In absence of inducer, the regulatory gene will undergo transcription to produce one repressor protein. This repressor protein goes and binds on the operator. Okay, and these structural genes are switched off. This much only you have to know. Now let's see what happens in the presence of inducer. In the presence of inducer, that is, inducer is lactose. This one which is making no. Okay, one point I forgot to tell. Completely it will not switch off. Little bit work will go on. Okay, it will be completely not switch off. It will work in minute amount because they want this enzyme later, like permease, beta galactosidase, and transacetylase. Why the permease is required? To bring the lactose in. So if this was completely switched off, there will be no permease. So they will make this active but in minute quantity. So permease is required to bring in the lactose. Now in second case, there is presence of lactose. So lactose has to come in. That lactose which is coming in is because of this Y, which is permease. See what happens here. See, this lactose. What is the function of this protein? It will go and bind on the operator and it will switch off this working. But what this lactose will do? Lactose will bind on the repressor protein and this repressor protein cannot go and bind on the operator. That's all you have to know. This lactose which is there will go and bind on the repressor protein. Repressor protein cannot bind operator. So what will happen to this gene? They are active now. So then they will produce beta galactosidase, permease, and transacetylase for the lactose metabolism. Beta galactosidase converts the lactose to glucose and galactose. Permease allows the lactose to come in freely, and transacetylase adds acetyl group. So this is about lactose. Then here lac operon. Glucose and galactose cannot act as inducer. Glucose and galactose cannot act as inducer. This lac operon will work only if there is presence of lactose. If you give glucose and galactose, this lac operon will not work. But if there is already glucose, there is no need of this lactose at all. Because what is required is source of energy for the E. coli. Okay, this is present in E. coli bacteria, lac operon. So source of glucose is there, then there is no need of lactose. Lactose will be used only if the glucose is not there. And that time this lac operon will work. Regulation of lac operon by repressor is called as negative regulation. Okay. So the first thing which we saw, this is negative regulation because there is a repressor protein which does not allow the functioning of the genes. That's why it is called as negative regulation. And the second point, regulation, lac operon is control of positive regulation. So lactose, when it gives these genes to work, it is called as positive. So this is all we have to see. Next class we'll see human genome project. In this, it is just like by heart, not much is there. So you'll write this.
So from which chapter those questions are?
This chapter only.